for our continuing use of the natural resources of their ancestral homeland. And we thank you all for tuning in what can only be called interesting times. It's a thrill to be able, nevertheless, to invite you to these virtual editions of our programs and to use live streaming to build a kind of digital stage serving Town Hall's community of curious and engaged Seattleites. Like everyone who's willing to stare or talk into their computers right now, some for the first time, though not, I should note, tonight's speaker, I'd like to thank Dr. Shubin for helping us keep the conversation aloft here at Town Hall. Tonight's program can be viewed in its native habitat here on Crowdcast, as well as on our Facebook and YouTube pages. Viewers who want to watch with closed captioning should go to YouTube, where you can enable real-time captions with the CC button in the bottom right corner of the player. And the video will also be available for rewatching immediately following tonight's broadcast. Tonight's presentation will last around 30 to 45 minutes, 30 to 48 probably. Afterwards, he'll take your questions. Please submit your questions using the Ask a Question button on Crowdcast, and Dr. Shubin will then step in to take them at the very end of his talk. We can't guarantee he'll get to every question, but we'll try to get to as many as possible. So keep them succinct to maximize your time. Upcoming programs around science and society include a look at the latest from the Hanford nuclear site, featuring Attorney General Bob Ferguson, Tom Mueller, and Tom Carpenter. Anthony Townsend on the future of driverless cars, I should say the imminent future of driverless cars. And our annual Engage UW Science Series, which is features reports directly from UW Labs by grad students engaged directly in work there, as well as appearances by Michael Shermer, Jillian Andrews, that's not true, Kirk Bloodsworth, Sister Helen Prejean, and tomorrow evening's program with Samantha Irby and Angela Garbins. We're adding new events nearly every day, as well as new events being released in podcast form. In fact, one yesterday and one tomorrow. And many of our past talks are available in video or podcast form on our digital media library. So in short, poke around our media tab on our homepage, and over the coming weeks, Town Hall will provide not only ways to stay plugged into the present, but plenty more rabbit holes for you to climb down from our recent and even distant past. Town Hall's programs are made possible through your support and the support of our sponsors. In particular, our Arno Matulski Science Series is supported by Microsoft, KUOW, the Wyncote Foundation Northwest, and the taxpayers of Washington State. But as most of you know, Town Hall is first and foremost a member-supported organization, and I want to thank all of our members watching tonight. You also likely know all those rumors about the vulnerability of nonprofits right now, they're absolutely true. Town Hall, like other organizations, is under significant strain, and so we hope you'll consider a gift at this time by becoming a member yourself through our website or by making a donation using one of the URLs in the comment threads of the respective feeds tonight. One final economic data point before we start, I promise. The fact that other beloved independent businesses are feeling the squeeze as well. And if you're interested in deepening your understanding of tonight's subject, and I certainly hope you are, since that's the thing that's brought us together here, and along the way in supporting entrepreneurs like our wonderful partners at Third Place Books, may I politely suggest that you use the Buy the Book button on our event page or the URL in the comment threads, rather than, say, turning to the local retail behemoth that used to specialize in books. Town Hall, after all, hearts Third Place Books. All right, then. Dr. Neil Shubin is a paleontologist, an evolutionary biologist, and a popular science writer, as well as the Robert R. Bensley Distinguished Service Professor at the University of Chicago and the Associate Dean for Academic Strategy of the university's Biological Sciences Division. Shubin is known for his landmark research on the evolutionary origin of anatomical features of animals, derived from fieldwork in much of North America, Greenland, China, Africa, and Antarctica. He has discovered some of the earliest mammals, crocodiles, dinosaurs, frogs, and salamanders in the fossil record. And one of his most significant discoveries, I should have gotten the pronunciation. Dr. Shubin, will you help me out with this? A 375 million-year-old fossil called
Tiktaalik rosea.
Thank you so much. Is considered an important transitional form between fish and land animals. Beyond the research itself, he is committed to sharing his work with the public through a variety of means. His lab maintains an active presence on Facebook and Twitter, and he takes it considerably further by sharing his discoveries, his knowledge, and his passion in terrific books, including the best-selling Your Inner Fish, A Journey into the 3.5 Billion-Year History of the Human Body from 2008, and 2013's The Universe Within, The Deep History of the Human Body from 2013. At least one of those other programs brought a visit to Town Hall, and his latest book, Some Assembly Required, Decoding 4 Billion Years of Life from Ancient Fossils to DNA, is the occasion of tonight's virtual return. Please join me in welcoming Neil Shubin.
Well, thank you. It's a delight to be here with you tonight, and to talk about Some Assembly Required. First off, I hope this finds you and your loved ones well and happy and thriving. And I look forward to the next 30, 40 minutes to talk about evolution and the great transitions in evolution. So I've been a paleontologist and biologist working on evolution for over 3 decades. It was kind of stunning when I realized that it's been over 3 decades, but there you go. And I began as an expeditionary paleontologist. I, wanted to go out and find fossils that tell us about the great steps in evolution. How did fish evolve to walk on land? How did mammals arise from reptiles? How did frogs evolve to jump, and so on? And, I was on my first expedition, one of the first ones I ever led. This is in the sort of mid 80s. And I got back from the expedition thinking, I'm going to be, this paleontology thing's going to be great. And a fellow graduate student left on my desk a pile of papers from molecular biology showing genes that build bodies and how those genes evolved. I'm like, okay, I got to learn this molecular biology stuff, because it's like the species I study in the distant past. I either have to change or I'm going to go extinct. So what I did is I became also a molecular biologist. And so my laboratory, if you were to come to Chicago, where I'm right now, is both finding fossils, going expeditions around the world, as you heard, from Antarctica to the Arctic and so forth, but also molecular biology, looking at DNA and trying to use both those approaches to ask the question, how do the great transitions in evolution happen? How did fish evolve to walk? How did the descendants of fish evolve to walk? How did birds evolve to fly? I mean, these are the great questions that have puzzled scientists and actually captured the imagination of scientists and the public for over a century. And so that's what really Some Assembly Required is about. And I was thinking of the book, and as I was finishing the book, actually, I was reading a biography. I'm going to share my screen. I was reading a biography of Lillian Hellman. Now, Lillian Hellman, let me share the screen here. Lillian Hellman had a very hard-living existence. As you can see in this slide here, this is a woman who was a fabulous playwright. She had a hard living. She drank, she smoked, she had amazing tales of a long and complex life. She was pulled in front of the House Un-American Activities Committee, but in front of Joe McCarthy's committee in the 50s. She was actually blacklisted from Hollywood, from Broadway as well. And in thinking about her life years later, during her autobiography, which I was reading as I was finishing Some Assembly Required, she had a quote. And she had a quote that captures the spirit of the book in a nutshell. And the quote is this: "Nothing, of course, ever begins when you think it does." I remember reading that quote thinking, wow, that's the book in a nutshell. Like evolution. When you look at the history of anatomical features, of structures, of organs, even of DNA itself, of genes themselves, nothing ever begins when you think it does. If you think that lungs evolved when animals first evolved to walk on land, if you think that feathers arose as the ancestors of birds took flight, you'd be in very good company. You'd also be wrong. And we've also known much of this for over a century. And that is the story in a nutshell, whether it's genes or anatomical structures. The changes that we associate with the great transitions in the history of life arose well before in a different context. And so much of evolution is about repurposing. And this story gains new meaning as we think about DNA and linking our studies of DNA with our studies of fossils. And it's really amazing. When you think about the molecule DNA, look at this here. Okay, what we have in DNA is a molecule that's in us. It's the double helix. This was very, very famously described. The double helix is about 6 feet long in each cell. That is, you have a 6-foot-long strand of DNA rolled up in on itself inside each cell. And not only inside each cell, inside the nucleus of each cell. Think about that for a second. We have about 4 trillion of our own cells in our body. Each one of them has a 6-foot-long strand of DNA rolled up inside itself, inside the cell. If you were to take all the DNA of our body, lay it end to end, okay, each one of those 6-foot-long strands from all 4 trillion or so cells in our bodies, it would run, and laid it end to end, it would run from here almost to Pluto. That's how much genetic material we have inside of us. And we've lived in an era of genome projects and genome technology. It took several decades to get the human genome sequenced. We can now get a rough copy of a genome sequence in an afternoon for less than $1,000, whereas before it was several $1 billion. We now have genomes for creatures as distant as from humans to corn, to flies, to worms, to thousands of species. We can do genomes in an afternoon, as I said. With all that genetic information and all that genetic and computational tools to analyze it, we now see DNA as a highly active molecule. It's crumbled up inside the nucleus of each cell, but it opens and closes as genes become activated in different ways to make different tissues. It's marvelously complex, and the origins of different genes are amazingly wondrous. And I'll start with one example, then we'll back off and we'll start to look at other aspects. Let me give you one striking example about some hidden mysteries of our own DNA. And it begins with this neuroscientist, excuse me, Jason Sheppard. Now, Dr. Sheppard is a professor of neurobiology at the University of Utah. And he's interested in the genes that control the formation of memories in humans, and uses mice as a model to study them because we have very similar genes in this regard to mice. And so he's interested in one particular gene called ARC, A-R-C. Now, ARC's an interesting gene. Mice that have a mutation in ARC have a problem. That is, they can solve a maze, but they don't remember their solution the next day. So in mice, it's clearly a gene that when it's disrupted, it disrupts memory, so it plays a role in memories. Humans that have mutations in ARC also have many cognitive deficits, as well as schizophrenia and many other mental disorders. So it's a really, really important gene for understanding the function of our brains, and particularly memories. So Jason was studying ARC. And like any good biochemist studying a gene and interested in neurobiology, he was studying the protein that the gene makes. And what he did is he popped it under a microscope. So he isolated the protein. This is what he got here. Jason sent me a slide for me to show you. And this is what he saw under a slide. He looked at this under an electron microscope. And what he noticed was where this arrow is pointing, you can see these little clumps, these little globules, balls that you see around. You can see them throughout the slide. Now, this is at super high magnification, okay? And he was looking at this and he was thinking, I've seen these clumps, these balls before. In fact, they're identical to something else I've seen before. And he remembered his training as a biologist, and he pulled out a textbook, in fact, a virology textbook, kind of relevant. And you could see what the arrow is pointing to there. He could see these clumps too. They look very similar to those of ARC, only this is not a memory gene ARC. These are clumps formed by HIV, the virus that causes AIDS. So what Jason decided to do is, okay, I'm going to get some expert opinion here. So he's in a medical school, University of Utah. He calls colleagues over in the next building who work on HIV, and he invited them over to the lab, and he asked them to look at his slide, and he did not tell them what was on the slide. He said, just what do you see here? And they're like, look down the microscope. They say, oh, that's HIV, the virus that causes AIDS. And Jason's like, nope, it's a memory gene, ARC, in mice and humans. Okay, now that set off a chain reaction of research where the teams sort of linked their resources to see what's going on with ARC. And they looked at the sequence of the protein of ARC, they looked at the gene of ARC, and it turned out that ARC, this memory gene in mice and people, is a repurposed virus. It has the signature of a virus of the kind of which HIV is as well. And when they looked at comparatively to it, they came up with the hypothesis that the way ARC arose is it was originally a virus that invaded the genome of one of our distant ancestors, entered the genome, and then as it entered the genome, it was repurposed by our distant ancestors. Instead of infecting and causing distress or disturbing things, it was repurposed to function in memories. Now, what these clumps do in HIV is really interesting. In HIV, these clumps actually protect the genetic material of HIV as it travels from cell to cell to do its work, right? To do its evil business, right? In ARC, it's actually a similar sort of thing. It's repurposed it because this capsule protects the genetic material as it goes from cell to cell in making memories and in serving in neural functioning as well. So it's really remarkable how a virus that entered the genome was domesticated or repurposed for a new use. So here's the origin of a whole new gene that came from a virus from outside the body. And it turns out that's not unusual at all. If you look at some of the proteins that are involved in pregnancy, such as some of them that are in the placenta, those were also repurposed viruses. And the list keeps on growing as we understand more about the genome. It's really remarkable. To the point where, if you look at our genome, 8% of our genome is derived from ancient viruses. 8% of our genetic material is ancient. It is a derivative from ancient viruses. If you look at our genome, though, only 2% of it are our own genes. Only 2% of our genome encodes for proteins. So we have 4 times more genetic material inside our genome than our own genes that are derived from viruses. So it's 8-2. It's really, really remarkable. It's truly remarkable. And it serves in everything from memories to pregnancy and in many other functions as well. So this just goes to show the mysteries inside our own genome. It's truly remarkable and very surprising. And this is all because of new technology allowing us to see evolution in whole new ways. And in fact, some of the most important new ways where we can look at evolution, the most exciting new ways, relate to this puzzle. This is one of the greatest puzzles of biology, and one of the great puzzles of biology that we've seen some significant progress in the last 30 years. What you see is a fertilized egg, right? So sperm, egg come together, they make a single cell. Now that single cell, from that single cell will be derived as it divides over and over again to go from 2 to 4 to 8 to 16 to 32 and on and on and on. It's going to eventually end up as a 4 or so trillion cell body, like you and I and everybody in this audience is today. We're 4 trillion cells with those cells all packed in the right way. They're differentiated from a single cell to become muscle cells and nerve cells and bone cells and skin cells and all the diverse cells inside of us. We call this process of going from a single cell to a 4 trillion cell body, we call that bodybuilding, right? And if you look at this slide, it captures one of the greatest puzzles of biology. How do you go from the single cell on the left to the creature on the right? And in that, what we have is an enormous number of discoveries that have revealed the inner workings of the egg and how DNA acts to build bodies. What we've done is we've begun to understand, say, the genetic recipe, the genes that interact to build the body, to control the number of cells and to control their differentiation and where they are and so forth. It's a really remarkable thing. And as we begin to understand this process of going from egg to adult in different species, we can begin to understand evolution in a whole new way, because changes in this process, say changes in the process of how you build bodies, explains a lot of evolution. Changes in the process of how you build the body of a fish can produce the body of an amphibian and so forth. So changes to this development, to how eggs go to adults, is a big piece of understanding evolution. And honestly, people in our field have been interested in this for over a century. And I want to talk about this historically a little bit, and then we'll go into some modern research. People have been interested in embryology and understanding, this transition from egg to adult and evolution for, well over a century. And one of the sort of apocryphal moments in this is the work of Auguste Dumariel. And you can see Dumariel pictured here. Dumariel had one of the world's greatest jobs at the time. So we're looking at Dumariel is right after Darwin published the first edition of Origin of Species in 1859. This is about when this portrait of Dumariel was drawn. And Dumariel was a really lucky man as well. He was the keeper of reptiles and amphibians at the Museum of Natural History in Paris. It's a great job at the time because people were coming back to Paris, from different parts of the globe with creatures that they found, creatures that they discovered. And he got to study them for the first time, to describe these species and to understand their basic biology. And he, one day, he received a package from people who were working in Mexico. He received a package of 6 of these salamanders. And they were working in Mexico, and they found these salamanders. These are salamanders, I'd say, about 4 inches long, 4 to 6 inches long. So they're fully functioning, sexually mature adults. So these are sexually mature adults, salamanders. But they captured the imagination and the interest of these collectors for a couple reasons. The reasons are, look at the gills. They have external gills. Look at the tail. It's a big aquatic tail. Here is a creature, a salamander, that seems to be sexually mature and fully aquatic. They thought, well, Darwin just published his Origin of Species. This is right after 1859 when Darwin published his book. They thought, well, we'll give these to Dumariel. Maybe Dumariel can understand how, the transition from life in water to life on land by understanding these creatures. Dumariel agreed, and what he did is he kept these salamanders. Little did everybody realize that the indigenous people of Mexico actually knew these salamanders extremely well, so much so that they had a soup recipe of these things. But they also knew quite a bit about their biology as well because of that, because of their local knowledge. Anyway, so Dumariel kept these salamanders. And he put them in his menagerie and he let them go. And, the salamanders are easy to care for. Just set up an aquarium, and a little terrestrial piece there as well. He left them some food and he let them be. And he came back and he noticed about 6 months later, he was in for a surprise. 6 months later, he looked in his enclosure, and these are the original diagrams actually from Dumerel's monograph. He saw these salamanders living with them as well. These salamanders just appeared out of nowhere, but look at them. They're fully terrestrial. They don't have an aquatic tail. They don't have external gills. In fact, their head's entirely different, very different shaped. It is like an entire different genus of salamanders. It's like somebody put chimpanzees in a cage one year and then came back the next year to find both gorillas and chimpanzees inhabiting the same cage. This got Dumerel's attention in a very big way. And he's like, wait, these new species, just new genera, this is a whole new different kind of critter, don't appear out of thin air. So what he decided to do is to study their biology, how they developed, how they go from egg to adult. And what he did was this: he found something really remarkable. He found that, like any good salamander, they start as egg and they hatch from their eggs, and they have larvae. You could see it at the top. The larvae have external gills and a big fleshy aquatic tail, and the larvae live in water, like little tadpoles. And they swim around and they get bigger and bigger and bigger and bigger. But then there comes a point when there are two possible states here. He noticed that the salamanders that are the big, fully aquatic ones, they just get bigger and bigger and bigger and bigger and become sexually mature with all those aquatic features. But then there's another way they can develop. That is, they can undergo metamorphosis through an external environmental trigger. They get a thyroid hormone is released, and they then lose the gills. They change the shape of the head, and they change the shape of the tail, and it becomes a fully terrestrial. So here's one developmental program that, depending on the environment, can yield two different states. So two different kinds of salamander can come about from the same developmental program. What's important here to realize is that a simple shift of development can bring changes across the entire body. Think about that. A simple shift in whether you metamorphose or not, or whether there's a hormone secreted or not, can produce changes to the tail, the head, the limbs, which I didn't talk about, the gills, and so forth. So this was really a critical discovery in sort of igniting people's interest in embryos, in studying embryos and evolution. And there's a whole tradition of people studying embryos and evolution in this way, to look to see how small changes in development can bring about big changes in evolution in the adults. Now we have we can relate this to our own bodies. If you look at people, these creatures here are human beings. Believe me, they are. And as human beings, we share a number of characteristics with other creatures. We are vertebrates. That is, we have backbones, we have skulls. So we share all these features with fish and birds and mammals and reptiles. Okay, these are what makes us vertebrates. Turns out there are other features as well that we share with them. We have a spinal cord. We have gill arches and gill slits that form in development. And those are not in the skeleton, but they're clearly in our development, in our development in our biology. For a long time, people have asked the question, what are the closest invertebrate relatives to vertebrates? That is, what are the closest relatives of squishy things to us? Clams, worms, you name it. What are they? Well, people studying DNA have come to the conclusion, well-supported hypothesis, I should say, that our closest invertebrate relatives are, get ready, these sea squirts. Look at them. Compare the creatures on the left to the creatures on the right. You're not going to see a whole ton of similarities. The creature on the right is sessile. It sticks to the rocks. It pumps water through an in-current siphon and an ex-current siphon. There's no apparent spinal cord. There's no apparent gill slits. There's no apparent body. And some of them get even weirder, right? Some of them like a little clumps. Well, what do? Well, guess what? Just like Dumerel, you got to study the development, how they go from egg to adult. And this is where it gets really, really, really interesting. You want to see the larvae of these things? This is the larvae of these things, of our closest squishy relatives. Look at it. It hatches from the egg, and it has a larva. And it has a larva with a head and a tail. If you look at the head, what does it have inside the head? Gill slits, just like our embryos. If you look at the rest of the body, it has a nerve cord, which becomes a spinal cord, just like us, and another structure, a notochord, which is also part of our shared history of things. So our closest relatives, their adults don't look like us, but their embryos sure do. So if you look at the life history of these things, what you'll see is they begin as hatch from the egg, begin as a tadpole, sea squirt tadpole you can see here. And they have that nerve cord and that thing called a notochord, and they have gill slits, and they swim around, and eventually they attach to rocks. And you can see that as you proceed, They attach to the rock, and as they attach to the rock, they lose everything that they share with us. They lose the tail, they lose the notochord, they lose the nerve cord, they lose the gill slits until they become that sessile clump. So the way evolution worked here is just like in the salamanders, it stopped early. That is, we are basically, our ancestors basically were a version of a sea squirt tadpole that kept the larval stage, didn't undergo all that other stuff, but we kept the larval stage, and that became an adult for our ancestors to evolve. So essentially, we are very similar to the larvae of the sea squirts. So eventually, if you keep the juvenile stage and just make that bigger, then you have something that's like our ancestor. Again, like doom reel, enormous amount of changes and surprising ones from looking at development. Now, we now have tools to look at this through DNA. That is, we can now manipulate DNA using techniques like CRISPR-Cas genome editing, which I'm sure you may have read about, to begin to see how DNA controls development. And what it does is it develops as it forms bodies and how it can evolve. One of the great stories of this is from a creature called an amphipod. My colleague here at the University of Chicago, Nipam Patel, has studied them. And in fact, he began to study them for a lot of reasons. And they're tiny creatures. My kids call them jumpies. They live in the sand and they hop around. And you can see them here. The head is on the left, the tail is on the right. And look at all of them. They're called amphipods because they have lots of different kinds of legs. See the front legs there? You have the front legs that are sort of like lobster claws. Then you have legs behind them that face to the back. And you have legs behind those that face the forward. And then you have other wispy legs as you go further back. So he thought this is a natural sort of creature to try to understand how legs form and how genes control, what organ is where in the body and how those organs develop. And what he noticed is that there's a genetic address to which organ forms where in the body. Now, this is a little complicated, but let me work you through it. So here what we have is 3 genes, UBX, ABDA, and ABDB. And they're different regions of the body that have activity of those genes. So let's just look at the front part of the body, which is gray here, where you have the legs that face backwards. In that section of the body, which is shaded gray here, you have legs that face backwards. In that part of the body, you only have one gene of those genes expressed, UBX. And where you have UBX turned on, you have a backward-facing leg, where the red arrow is. Where the blue arrow is, you have forward-facing legs. And that's a part of the body where the 2 genes are turned on, UBX and ABDA. So there's a genetic address for each segment. Where you have UBX alone, you have a backward-facing leg. We have UBX and ABDA, shown in the hatched pattern, you have a forward-facing leg. And in fact, when you have ABDA and ABDB in the rear end, you have the wispy legs. So Nipam thought, well, what if I use this genome editing technique to manipulate these genetic addresses? Can I control what legs form in different parts of the body? And the answer is yes. What he did was, he made an embryo that doesn't have ABDA. So all it has is UBX in one area. So basically, he took out ABDA. See that? So he got rid of that hatched portion. And now look at the bottom. He now has, getting rid of ABDA, he has one whole region, which is just UBX activity. And all you end up with is backward-facing legs. So here he's controlling the genetic address simply by getting rid of a gene using this CRISPR-Cas genome editing technology and changing the organ that forms there. And he can do that almost at will. He can change it almost every single one of those appendages by changing the genetic address. It's just a remarkable set of experiments. But the story gets even deeper because it turns out that flies have these genes too. You see a fly embryo there where they're color-coded. I'm not labeling them, but color-coded. And what you have in these color-coded areas is the information, a genetic address for whether a leg forms in a particular area, a particular kind of wing, a mandible, or what have you. It turns out that we have these genes too. They're active. Versions of these same genes are active in the formation of our own body plan. And what are they doing? Well, they're active in a lot of different organs, but one particular place is in our vertebral column. That is, there's a genetic address for the different vertebrae in our vertebral column, whether they're neck vertebrae, the cervical vertebrae, thoracic vertebrae in the middle, the lumbar, sacral, and so forth. Well, you can ask the question, what if we do a similar set of experiments in mammals as Nipam did in his little amphipod? What happens if you change genetic addresses? Do you change the kind of vertebra that develops in different parts of the body? Well, let's look at this. These are experiments done at the University of Michigan a few years ago. Here's a mouse, cute little mouse there. And now we're just going to zoom in, if you will, at the rear end of the vertebral column. Okay, so look back there. That's what we're honing in on. okay? If you look at the normal development of a mouse, there's a genetic address for each kind of vertebrate. Look at the embryo in the middle there. You could see two genes that we're showing you. One called HOX10. These are versions of the ABDA and UBX genes you saw before, but they get a different name in mice and people. You see one gene HOX10, and you see another gene HOX11. There are three regions. There's a region that only has HOX10 activity. There's a region with the arrow where you have both HOX11 and HOX10 activity. And then there's a region further back where you only have HOX11 activity, right? So HOX10, HOX11 and 10 together, and HOX11 alone. Turns out that genetic address determines which vertebra forms where. Lumbar vertebrae form where you only have the HOX10 address. We have the joint address of HOX10 and HOX11 where that red arrow is showing. You have sacral vertebrae. Well, what if you do the same experiment that Nepom did? What if you get rid of HOX11 and just make only HOX10, right? If you get rid of 11, you only have a HOX10 address. What kind of vertebrae would you have? You'd only have lumbar, right? If the hypothesis is right. And that's exactly what happens. If you knock out HOX11 and you only end up with HOX10 addresses, boom, look where the blue arrow is, you get lumbar vertebrae where sacral vertebrae were before. So you can change the address of different parts of the body and change tons of different structures. So really, the genes that form much of our body, where did they begin? Well before the formation of our body. They are repurposed genes that build the bodies of flies, of worms, of amphipods like parahyali. So there's a commonality to the toolkit that builds us. Nothing, as Lillian Hellman said, ever begins when you think it does. And following that theme even more, let's return to my own research. This is something I work on. I look at the transition from life in water to life on land. You see the fish on top and the land-living animal, tetrapod, limbed animal on the bottom. Now, if you look at this, you could say, well, there's all kinds of things that have to change here for animals to, motor on land. You have to have a creature with a different shaped head, creature with a neck. No fish has a neck. Fish have fins. Animals that live on land have limbs, and that's just the tip of the iceberg. What about lungs? Think about that. You'd have to evolve lungs at the same time as all these other structures, and all sorts of other things have to go on for fish to be able to evolve on land. If you made a list of all the features that are needed for fish to evolve to walk on land, it seems impossibly long, and you'd naively think they'd all have to arrive at the same time, like lungs, right? I mean, how would you evolve lungs at the same time you evolve limbs? Turns out there's a long history here, and it's very, very, very surprising. And it relates to Lillian Hellman's quote, nothing ever begins when you think it does. Lungs. Takes us to one of the great characters in all of comparative anatomy and paleontology, Bashford Dean. Bashford Dean had two passions in life. He did most of his work in 1910, 1920. He had an incredible two jobs. He was a curator of vertebrate paleontology, of armored fish. That was his passion, armored fish. Ancient armored fish at the American Museum of Natural History in New York, but he also held the position of curator of medieval armor at the Metropolitan Museum of Art right across Central Park. He held joint curatorships at both institutions, the Art Museum and the Natural History Museum, literally right across the park from one another. In one case he was studying armored fish, in the other he was interested in armor. He just had a fetish about armor. And he used to walk around New York City with in samurai armor and in various helms and medieval armor as well, and he designed his own armor for World War I. He was quite the character. But he discovered that one important thing, that lots of fish have lungs. And in fact, lungs are the primitive condition in fish. It was really a remarkable thing. And he was sort of fascinated by these fish, which I say is the world's cutest fish. That is the Australian lungfish, Neoceratodus. Look at that thing. It's just gorgeous. And they live a long time. We had one here in Chicago that lived to 84 years in Shed Aquarium. And they don't know how old it was when it came here. They lived really long time. But anyway, lungfish have both lungs and gills. Both lungs and gills. And so they use the gills when the oxygen content of water is high enough. But when the oxygen content of water drops, what they'll do, and this is after one of Bashford Dean's monographs, they'll go to the surface and gulp air. They'll gulp the air and force it into their mouth, and then force it into lungs. And he looked at their lungs, and it turns out their lungs, and this is another lungfish, there's three different genera of lungfish. There's one in Australia, one in South America, and one in Africa. This is the African one. If you look at the lungs, you can see the lung here on top. You can see it's this long structure, and it lies adjacent to the gut tube. So look in the middle where the arrow goes. You have the gut, and then you have the lung, and there's two lobes to the lung. If you look at the lung, look at the bottom right, they even have structures like our own alveoli. it's really remarkable that when you look at the histological structure of the lungs of these fish, they have a histological structure virtually identical to our own. And in fact, if you look at the genes that control the development of air sacs in diverse fish, they're similar to the genes that are behind the development of our own lungs. So there's really a deep tie between these lungs and our own lungs. And the story is really rich, because if you look at the evolutionary tree of life, so what you have here is a very simple tree of fish and limbed animals. So on the right, you have a limbed animal. And the closest living relatives of limbed animals are lungfish. You can see them right there. And then there are other fish as well. As you go deeper in the tree, you have sharks and other fossil creatures. But if you look at the presence of lungs and just map them on this tree, where are they? Boom. Lungs are primitive. Lungs evolved eons before animals took their first steps on land. And so what you have is a situation where lungs were functioning to help creatures live in aqueous water environments where the oxygen content would vary. Turns out they already had lungs by the time they evolved to walk on land. And when you think of this transition, every other trait of this transition pretty much arose in fish living in aquatic ecosystems. My team and I spent about 6 years looking for a fish that was intermediate between fish and limbed animal. We were looking for a flat-headed fish with fins with arm bones inside, and that's what we found. After 6 years worth of work up in the Arctic, we found a flathead fish with fins with arm bones inside. Here's a fish with scales in its back and fins with fin webbing. You can see the fin on the right there. But like a limbed animal, has a flat head with eyes on top, has a neck. You see the head is separated from the shoulder girdle, can move independently. And when we opened up the fin, it had bones that correspond to upper arm, forearm, even parts of a wrist. It's really remarkable. So what does this mean? This means that fish living in aquatic ecosystems in the sort of late Devonian, let's say about 375 million years ago, already had lungs, already had arms, already had wrists, already had necks, already had a head that was, like a limbed animal, such that when the time came for them to walk on land, they already had every tool. They were repurposing what already existed. All the features that were associated with the invasion of land originally arose in fish living in aquatic ecosystems. Now, this is not just about these guys. We can relate this to every other transition in the history of life. Take birds. Here's a gorgeous bird. Look at that. But think about birds. They have all kinds of specializations for flight. They have wishbones. They have wings. They have feathers. They have hollow bones. They have high metabolisms. And the list goes on and on and on. Guess what? We find each one of those features in dinosaurs, in feathered dinosaurs. Dinosaurs which have feathers, dinosaurs which have a wing-like structure, dinosaurs which have reduced toes, wishbones, and the list goes on and on. And these weren't flying. They were using these traits to capture food on land. Feathers were originally not serving to function in flight. They originally functioning for thermoregulation as insulation or for courtship displays and things like that. So just like lungs, feathers, and other structures, all these things, where the major step in evolution is repurposing what exists. Nothing, of course, ever begins when you think it does. It begins earlier in a different context. And in that context, what you find is that structures arise doing one thing, and then they repurpose for another. So much of evolution is not around evolving new structures per se. It's finding new functions for old structures. Thank you very much. I'm more than happy to take your questions, and if you've typed them in, I'll answer them as we go. So I see a number here, so I'll just dive in. Okay. If the DNA, so I'm just going to read them, and so 2 votes. I'll just go. Would you be willing to wade into your opinion of the debates about group selection, multi-level selection, as some evolution call it? Yeah. So group selection. So this is a debate that's going on for years about, particularly about altruistic traits. So if there's a trait that appears to hurt the individual but appears to be good for the group, how does it come about? So there are 2 notions of how these kinds of traits come about. One is perhaps there's the groups that have individuals that sacrifice themselves for the benefit of the group. Maybe those groups will be better and those features will go. The other is maybe it's that these traits have other effects on other individuals, so that what may appear to be altruistic actually helps me because it helps me because I share genes with the individuals that I'm helping or saving. So there's the hot debate, because the idea is group selection is actually a very slow process, whereas the individual selection is a very efficient process. And so in most cases, people have tended to favor the explanation that it's not group selection that's important here, but it's more individual selection. But that being said, there are many cases where as you put it, multi-level selection can come into play. That is, you can have traits that act at different levels that can help the individuals that have them because they're members of a particular group. So cooperation, for instance, can have different origins here. So when you think of traits like sexual reproduction, for instance, that's probably group selectionist explanations are the ones I would personally favor. Now, there'd be a lot of people would sharply disagree with that, but for certain traits, I would certainly, particularly sexual reproduction, because it increases the diversity. Creatures that have that are more evolvable in certain ways, and groups that have that are more evolvable in certain ways. So that's one of the cases where I would favor group selection. But it'd be topic by topic. So second question, on the topic of genetic addresses, do you believe developments in this manipulation can pave the way for man-made species? Yeah, it could. Let's put it that way. Should it? Probably not. But you could manipulate species to be reproductively isolated from other ones. But I don't know why we do that necessarily. Maybe for agriculture in some ways. Maybe we're already manipulating plants and certain other kinds of creatures, microbes and others, for particular purposes, to clean the environment, to detoxify things. Plants, obviously, are for genetically modified organisms, for agricultural purposes and so forth. So we're already doing a lot of that, quite a bit, to be quite honest. And CRISPR-Cas actually makes some of this much more efficient. But we've already been doing it through other means, through breeding, just animal and plant breeding, to be quite honest. Did I answer the whole question? I think I did, yeah. COVID virus. Any truth to the rumors that blood type and/or sex or any other genetic factor affects susceptibility? Honestly, I don't know. This is a fast-moving field, and I'm not an expert in it, so I'd be unwilling to sort of offer an opinion because it's sort of outside my zone. And by the way, just to talk about COVID virus for a second, we're witnessing science happening in real time. We're watching science that normally happens in months and years. We're watching it happen in days and weeks. Just look at, follow it in the news or on Twitter or what have you. And it's also happening very fast because we now have a different kind of scientific publication process than we had in biology before. We have what's called bioRxiv and medRxiv. What you have is people can post papers online before peer review. So the results get in all diverse, scientists' hands before they've been peer reviewed. So what you're seeing is result after result after coming out on SARS-CoV-2, the virus, and COVID-19, the disease. You're seeing it come out very rapidly. That's one of the reasons, since it's coming out rapidly, it's not peer reviewed, and people are doing their best with limited data. What you're finding is there's a lot of discrepancies that are out there. How many asymptomatic cases? Who's susceptible? What surfaces are most likely to, and how long can the virus hang on surfaces? Simple questions, which are very complicated when you try to get them scientifically. You see a diversity of viewpoints here. And so the important thing is when you look at results for the virus SARS-CoV-2 or the disease COVID-19, keep in mind that you're looking at science happening in real time and you're looking at hypotheses. And really what we need is better evidence. And that's what people are trying to do. And by the way, we're getting evidence pretty quickly. But just stay skeptical about anything you see right now, because again, the evidence is coming in. We're just in the early. Really, we've only been in this for 2.5 months, and it's amazing what we know in 2.5 months. The gene sequence. The sequence for the virus came out in mid-January sometime, early mid-January, and so much has happened since then. So this is an interesting time for science and for the science and for the public to see science, because you're looking in the hood of science and seeing it happening in real time. So that's a long way of saying I don't know the answer to that, but it's also a short way of saying I don't think anybody else does yet either. And if they do, come back in a month or two, see what the later experiments show. Also, just briefly, I would love to hear about the emotional process of seeing a mod of a beloved video game. So basically, what happened? Seeing my discovery. So. We discovered Tiktaalik rosea. Here's a plushie of Tiktaalik rosea. Somebody sent this to me. I get all kinds of plushies sent to me. A Darwin plushie sent to me. I got Tiktaalik plushie sent to me. This is the creature I showed you before. It's a cast, an unpainted cast of the head. This is it here. The one that has the fins with the arm bones inside. Yeah, one of the greatest things I saw was a Minecraft mod. Everybody knows Minecraft now. A Minecraft mod of Tiktaalik. Yeah, I hope it's a friendly, docile creature. That would make me happy. If the DNA of sea squirts was sequenced, would that allow us to test the hypothesis sea squirts are our closest invertebrate ancestors? Well, yes, it has been sequenced, and sea squirts are, or they're that group of creatures, Urochordates, they're called, are our closest closest relatives of vertebrates are Urochordates, which is pretty cool when you think of it, because the adults look very different, but man, when you look at the embryos, the larvae, it's amazing. Do we know how, for example, the HOX10 and HOX11 get into the segments Does something diffuse across cells? Does it follow dividing cells? Where some cell ancestor divided in two. What controls the activity of these genes? Well, what controls the activity of genes is very interesting, because what you have are networks of genes interacting with one another in each body segment. And what you'll have are factors that cells secrete that diffuse across the embryo. And you have cells dividing at the same time. So take these diffused factors that go across the embryo, take cell division, and then take networks of other genes that are being turned on and off in response to those activities. Then you get patterns of gene then that controls activities of genes like the HOX genes and so forth. And so, yeah, there are genes upstream of HOX which control their activity, and there are genes upstream of those which control their activity as well. And it's there's a relationship between the proteins that cells secrete and the genes that are active in cells, the cells that are dividing, and their position in space. All that comes together to control gene expression. And then you have the chromatin, that is the balls of DNA as it's wound up in the nucleus, opening and closing, controlling the activity of genes. It's an amazingly beautiful dance, and we're just beginning to understand it really at a molecular level. Why are octopuses, you're right, you did it right. Why are octopuses so intelligent? They have really big brains. And those brains, there's a team at the number of teams trying to understand consciousness in octopuses, because they can solve puzzles. They can, the famous, I think it was in Seattle, wasn't it, where they put sharks in the tank and they would disappear, and it turns out there was an octopus that was eating the sharks every night or every week or whatever. I don't know why they're so intelligent, but their brain to body size ratio is really huge, so that certainly contributes to it. But how it's wired, I don't know. I think people are studying that right. I know that's being studied pretty actively right now at Woods Hole in the Marine Biological Laboratory. If a genie granted you the ability to have one question you are researching answered, what would that question be? I would like to know how, I'd like to know how cellular life originally came about. Because we have lots of ideas, we have lots of experiments, but I'd love to know how that happened. When you think of cellular, I can imagine many working hypotheses how replicating molecules can happen. But a cellular life with a membrane, a metabolism, and so forth, I'd love to see the steps involved in that, because it's going to be repurposing. It's going to be Lillian Hellman. It's going to be nothing ever begins when you think it does. I know that. But what are the steps of that? That would be really cool to see. Creationists often cite the eye as something that would have to have been created. But isn't there genetic continuity back to light-sensing cells and primitive creatures? That's correct. Yes, there is you can follow, from primitive photoreceptors, actually from primitive photoreceptor molecules and opsins and things, you can go all the way to eyes, and you look at the comparative record. If you look at our eye, it's a camera eye, and then there are more simpler versions of the camera eye as you go to other creatures. And also, when you look at the genes that control the development of eyes, whether it's flies or people, there's a similar sort of genetic network that gives rise to eyes as well. So you can look at the evolution of eyes at multiple levels. You can look at it at the level of the organ itself, right? How it's structured with lens and retina, and see and trace that comparatively to different creatures. You can look at it at the level of the proteins that control photoreception, the opsins themselves, and how they duplicate and how they control how we perceive different colors. And you can look at the genes that control all that stuff, right? And each one of those are showing the continuity that can happen with the origin of eyes. How did bones evolve from those closest invertebrate ancestors? That's a good question. Now, skeletal systems, we know, have multiple origins in different creatures. So some skeletons, like are calcium carbonate based, like shells and things like that. Others are hydroxyapatite based, like our bones and things like that. But when you look at the first skeletons, skeletal tissue. It's an open question whether the first skeletal tissue in vertebrates like us were teeth or bones. Because when you look at the first bones, the first hard parts, they tend to look a lot like teeth with an enamel layer on the outside and a dentin layer on the inside. And it turns out that the first skeletons we see are armor on the outside of the body of early jawless fish. You can find them. They're fairly common in the fossil record. And when you look at that armor on the outside of the body, and you look at it under a microscope, it's the structure of teeth. It has the enamel on the outside and dent in the middle. And so the argument goes that the first skeletons came about as like a tooth-like developmental system that was on the outside of the body. And then later that whatever the developmental process was that led to that coating on the outside was redeployed to make teeth on the inside. So we see armor before teeth. And so that's called the outside-in hypothesis. So it's actually a very interesting. And in fact, if I can have that genie back, I'd like to see that too. That'd be kind of fun. Maybe can the genie give me two? If I ask for like the genie for two things, that would be another one I'd ask for. Is there any evidence for the original emergence of consciousness? Oh, good question. Well, I mean, embedded in that question is also the notion: is consciousness unique to humans? And are there antecedents to consciousness that we see in other creatures? And I would say, as somebody who thinks about antecedents all the time, and you know where I'm going to go with this, I would say the answer is yeah, there are antecedents in other creatures. I would think that probably that cephalopods, that octopuses have a version of consciousness, and versions of consciousness we see in other creatures. I mean, the more we look, the more we find mental states exist in other versions of mental states that we used to think that are quintessentially human, that they're present in other creatures. There was a study out of the University of Chicago actually about 6 years ago now that showed pretty clearly that rats in a certain regime show empathy for individuals in their population that are suffering. And it's not just caring for them; it's actually empathy. And it was a paper published in Science by Peggy Mason. If you want to Google it up, and just type in "rat empathy science" and you'll get the paper. But that's been a number of studies. Just read. There's a wonderful book that came out last year by Frans van der Waal. I forget the title of it. It was a bestseller talking about just this issue. So yeah. About how versions of our own mental states are seen in the creature, particularly emotional ones too, but the consciousness is embedded in that as well. Following up on the consciousness question, does your line of study lead you to be vegetarian or more in that direction? Yeah, I flirt with vegetarian, pescatarian, vegan all the time. Yeah, I'm mostly vegetarian. In the field, I'm vegetarian. Actually, in the field, I'm vegan because one of our field crew members is vegan, and it's easy just to go, everybody go vegan rather than make two different dishes, that kind of thing. But yeah, I tend more to that. I mean, I'll go through stages where I'm straight on vegetarian, and then I'll add fish, then I'll stop and maybe I'll have a bird for every now and then I'll go back, and yeah, I go back and forth. But yes, I aspire to vegetarian, let's put it that way. How far back in time could this trend of biological repurposing extend? I would say all the way to the beginning. What does this mean for the origins of DNA? I would think, take it all the way back. I think nothing ever begins when you think it does. I think evolution, repurposing is it. So when you think about, there's a couple ways this can happen. So duplication, copying is one important way that new things happen. But once you duplicate, you can repurpose. Also recombining, that is taking two different parts, 1 + 1 to make 4. And that happens a lot. It's like, when, you had suitcases, we had wheels, all of a sudden somebody put wheels on suitcases and the world's changed, right? Well, the same thing's true with biology, when different parts come together. So repurposing, recombining, modifying, duplicating, these are all the pieces of the evolution of life. And I would say that they were part of it from the very beginning. All right, we're done with questions. This was really fun.
All right, well, thank you so much, Dr. Neil Shubin, for being here this evening with us. It was a fascinating presentation, and thank you everybody else for joining in this evening. If you enjoyed this event, you can find many more just like it on our website, townhallseattle.org. We hope that you will also consider making a donation to Town Hall Seattle, as your support will allow us to continue to provide events just like this one this evening's. If you're interested in purchasing a copy of Neil's book, Some Assembly Required, you can use the link on this live stream page to purchase through our friends at Third Place Books. And finally, thank you again for being here tonight. Stay safe and have a great evening.